Hello. Am I audible?
Yeah, you are.
Yeah, good evening, everyone, and welcome. Thank you for joining today's session. And I'm excited to kick things off by briefly introducing Boltzmann Labs. So, we're an AI-driven research company building an integrated platform for modern drug discovery, from designing proteins and antibodies to generating small molecules to enabling retrosynthesis. So, our goal is to make high quality computational biology and chemistry accessible, fast, and intuitive for teams across biotech and pharma. Yesterday we had a fantastic webinar focused on small molecule design, which was given by Catherine, where we explored how AI can accelerate early stage discovery. And today we continue the momentum with a session on enzyme engineering, which is going to be led by Srivani, our bioinformatics scientist, who has been doing incredible work in this space. And with that, I'd like to hand it over to Shivani to begin today's presentation.
Thank you. Good afternoon, everyone. I am Srivani. I work as Bioinformatics Scientist at Boltzmann Labs. Today, I'm going to give you insights about AI and enzyme engineering. One second. Okay, let's start something very basic. What is enzyme engineering? Enzyme engineering is a process of designing and modifying or optimizing the enzymes which you have in your hand to make it perform better than the natural ones which you get. Basically, there are three strategies for it, which is directed evolution, rational design, and machine learning or AI-driven enzyme design. So what is directed evolution? In directed evolution, we'll just introduce a random mutations and we'll create a library from them. After creating library, we'll look for the performance of those created enzymes and they select the one which has more performance. And then in rational design, we use the structural and mechanistic knowledge of the enzyme which we have in our hand. We'll understand the active sites or the role of the specific residues. Then we'll introduce precise mutations to improve their functions. So this is the overall thing that happens in the rational design. And in ML and AI driven enzyme design, this is the newest and fastest growing approach. AI models, they can analyze the large sequence database and predict the beneficial mutations, and even design a new enzymes from the scratch, which we could use later for our next research things. And why is enzyme engineering needs AI? You know, the traditional enzyme engineering has relied heavily on the trial and error. It takes like months and years to design something new. The main challenge is the sheer size of the search space. A single protein can have millions of possible mutations. To test them and experimentally validate, it's simply impossible. And also, the wet lab screening is slow, expensive, and often gives the limited insights. This is exactly where the AI makes the difference. AI can rapidly explore this huge search space, and also it scores the mutations, it predicts the outcomes in minutes that would take months and years in the lab. How AI helps in enzyme engineering? Uh, Basically, what AI does is it learns the patterns from huge data sets of natural sequences and uses these patterns to predict the functional or any other characteristics of the enzymes. It can estimate the thermodynamic stability, pH, and its solubility, hydrophobicity, etc. These properties it can predict. By doing this, AI reduces the experimental screening burden by like almost 10 to 100 times sometimes, and lets the scientists focus only on the most promising variants. Let's see how AI understands the proteins. The main thing is AI models are mostly trained on millions of natural protein sequences. After learning these protein sequences, they'll know the evolution and the conserved motifs that are present in the proteins, also their structural patterns and the residues which are responsible for their function, and stability as well. They learn which residues are rigid and also which form the active site pockets, which can tolerate the stress, and at which residues we could do the mutations. Using all this information, AI predicts which residue belongs to the binding pocket and which changes will impact their activity. Protein language models. These are some examples of the protein language models, which are ESM, PROT, T5, MSA, Transformer, Progen, etc. The modern PLMs, like these, treat the protein sequences just like a just like a sentences in a language. Like they'll learn the grammar which is present in the amino acids, the rules that dictate the folding or the stability which is responsible in the amino acid residues. Through these, they'll generate embeddings. Embeddings are nothing but the numerical representations of what they have learned. These numerical representations capture the structural tendencies, solubility patterns, or active site signaling, and evolutionary conservation. And these embeddings serve as a powerful input for next downstream prediction tools, enabling high accurate property predictions. AI can also predict the thermostability, solubility, active site changes, substrate preferences, etc. They can identify the mutations that improve the stability, regions that affect the solubility, and substitutions that alter the activity. It can also predict the substrate preferences by analyzing the pockets that are present in the enzyme or the protein sequences which you have given. Together, these predictions help guide the rational design and significantly improve the experimental success rates. Instead of doing those all these in the wet labs and which consumes years of time, you can use these AI models and could predict the functions, and also you can improve their functions, etc. Till now, we have seen the prediction models. Here, we can look into the sequence design models, where AI can actually design new enzyme variants. These models learn which residues contribute to folding or the which are responsible for the stability and suggest the mutations. Through these mutations, we could enhance the performance of the particular enzyme. They can even generate entirely new sequences within a protein family. But they'll preserve the global structure by improving desirable the properties which you want. So let's see how AI complements the wet lab experiments. AI doesn't replace the wet lab experiments, but it makes them far more efficient. It identifies the mutation combinations where can't even think of. It provides the statistics confidence and hypothesis driven design. Result is faster, cheaper, and more predictable discovery cycle. These are some of the examples. These have been made using our AI Boltzmann AI tool, wherein we have increased the thermodynamic stability, enhanced the catalytic activity of different enzymes like alpha amylase, superoxide dismutase, collagenase, and alpha galactosidase. There are many more examples which we have run in our lab. Now that we have seen how AI reads the protein sequences, how embeddings power predictions, and how AI accelerates enzyme engineering with the real examples from our Boltzmann tool, Boltzmann AI brings all these into one single platform, which is Boltzmann AI Discovery Suite. It combines the prediction, scoring, designing, and simulation, making the modern computational enzyme engineering accessible in a single place. Let's dive into that. Can you look at the screen?
Yes,
Hello.
Shivani. Yes,
Okay.
yes.
Okay. This is Boltzmann AI Discovery Suite. This is our home page. Here we have the AI agents. You could just search for something like pH and it will give us the tools. If you select something like enzyme pH prediction, it will directly take you to the tool where you can predict the enzyme pH. Also, if you want to search some random things, like if you want to know about enzyme like calbi, you could just search it by shift and enter. It will take you to our chat box. Here I have done something for you earlier. I have searched for the calbi and asked for research articles on that. It has generated the research articles here. And also we have the dashboard where it displays all your recent projects. And mainly we have three modules. One is for protein engineering, one is for small molecule design, and the other one is synthetic chemistry. We also have the documentation related to this. Let's mainly dive into the enzyme thing. Like we have tools to generate the enzyme, we have tools to predict the properties of this enzyme. uh, for this to generate the enzyme, if you even have the simple thing like the EC number of the enzyme, you could just directly type your EC number and you could ask for the number of sequences to generate. One second. Like you could see the results in many formats like grid table analysis, where you could see the each generated sequence along with its properties like this. From these, you could select your desired one based on the molecular weight or PI or hydrophobic count, and these specify the amino acid residues present in each sequence probability. For property prediction as well, we have tools like this enzyme property prediction. If you have enzyme sequence with you in your hand, you could just enter the sequence here, or if you have a substrate structure as well, you could just upload the file here and you could ask for to predict the pH or enzyme solubility or anything like that, and it will give you the results like this. Predicted pH and all. It also gives the EC number as well. For structure prediction as well, we have protein structure prediction, which is for single sequence. This is multiple bolts to is for docking, and we have separate tools for docking as well. Or if you want to do any mutations in the sequence in your hand, you could just use random control mutagenesis, where you could give the sequence and ask for it to generate the number of mutations as well. Also, you could protect the residues which you don't want to mutate in that particular sequence. Like if you know the if these residues are responsible for catalysis and you don't want to disturb it, you could just enter those residues here and ask for it to mutate. It will generate something like this. Mutated sequence length and its properties. There is another tool for the mutation as well, which is ML directed evolution, where you could just upload the sequence and ask for it to mutate the residues which you want, not the like the previous one where you protect the residues. Here you give the residues which you want to mutate, and it gives the results like this. We also have toolbox models like ligand MPN, and like if you have the backbone and the substrate as well and you want to build a conformation 3D conformational structure based on the ligand conformation, you could use this tool. We also have the dermal MPN, which tells us the thermodynamic stability in the mutated sequence in the point mutations exactly. This is the position where the mutation occurs. This is the natural residue which is present in that sequence, and and the residue which replaces the wild type one. Like here, methionine is replaced with alanine. Also, if we have only the sequence and you want to know its EC number, you could use this clean. We could briefly discuss this during the hackathon. We could briefly discuss it, all the tools there. That's it from my side. Does anyone have doubts? Hello?
Yes, Shivani.
Yeah.
Shivani, are you saying something?
No, no. I'm waiting for someone to ask doubts or
Yeah, if you have any doubts, you can ask in the chat box. You can also unmute yourself and ask. So, if there are no doubts, we'll wrap up the event.
yeah.
So thank you so much, Shivani, for that insightful session on enzyme engineering, and it was incredibly valuable, and I'm sure everyone here learned a lot. And to everyone who joined us today here, thank you for being here. And before we close, a quick reminder, our upcoming hackathon BoltzHack is open for registrations. And so if you haven't signed up yet, this is the perfect time. We also have exciting cash prizes, certificate of participation, and internship opportunities that we'll also release soon on our LinkedIn page. So we would love to see all of you take part in this hackathon. And thank you once again for joining and have a wonderful evening.